how are you? I hope you all are fit and fine at your places. So student, myself Priyanka Joshi in front of you. Let's start with today's biology topic. Students, in our previous video we have almost completed the chapter that is the fundamental unit of life. So today we are going to discuss the question answers, the exercise question answers of your Science NCERT book. So student, please listen the answers carefully and after that write the question answer in your fair notebook. Clear student? So students, in the chapter Fundamental Unit of Life, we discuss about the cell, the types of cell, prokaryotes, eukaryote cell. There are two types of cells in plant. It is plant cell, and in the case of animals, it is the animal cell. Here we discuss about the difference between the plant cell and the animal cell, and we have discussed about the different cell organelles, that is mitochondria, endoplasmic reticulum, Golgi apparatus, ribosome, lysosomes, that we covered these all topics in this chapter. Now today the discussion of question answer. Clear student? So here the student first question is, make a comparison and write down the difference between the plant cell and an animal cell. In this question you have differentiate between the plant cell and animal cell. So student, see, this column is of animal cell and that one is of plant cell. So the first point is cell wall. The cell wall is absent in case of animal cell and the cell wall is present in plant cell. Next is shape of the cell. So animal cell has rounded shape. It is irregular in shape, whereas plant cell has a rectangular shape. It has a fixed shape. Okay, so the next topic is vacuole. There are one or more small vacuoles are present in the animal cell, whereas in the plant cell only one large vacuole is present that occupy about 90% of the cytoplasm. Clear? Next is centrioles. Centrioles are present in all animal cell, whereas in case of plant cell they are present only in the lower forms of the plant. Centrioles are absent in higher plants. Okay. Next is chloroplast. Students, chloroplast is absent in case of animal cell. Clear? It is present in plant cell because plants have a autotrophic mode of nutrition. They prepare their own food. So chloroplast is very much necessary for that. Chloroplast is also known as kitchen of the cell. Okay, so the process of photosynthesis takes place within the chloroplast itself. So chloroplast is present in the plant cell. Next, plastids. Plastids are absent in the animal cells and they are present in the plant cells. Chromoplast, chloroplast, they all are come under the plastids. Okay, so the next is plasma membrane. In animal cell, only the cell membrane is present, whereas in case of plant cell, cell wall and cell membrane both are present. Next, lysosomes. Lysosomes occupies or they are present in the cytoplasm of the animal cell and lysosomes are absent in case of plant cell. Last topic is villi. Villi are present in animal cell and in plant cell it is is rarely found. Clear, student? So this is the difference between the plant cell and the animal cell. So the second question is, how is a prokaryotic cell differentiate from eukaryotic cell? Listen it carefully.
Difference between prokaryotic and eukaryotic cells. Prokaryotes and eukaryote cell differ in their structure. The major differences exhibited by these cells are mentioned here. So, in prokaryotic cells, they are relatively simple, small, ranging from 1 to 2 micrometer, whereas eukaryotic cells are complex, large cells ranging from 10 to 100 micrometer. Prokaryotes possess rigid cell wall which prevents rupture of the cell. Eukaryotes has no rigid cell wall in vertebrates, but in plants cell wall is present. Membrane bound organelles are absent in the prokaryotic cells. Membrane bound organelles are present in eukaryotic cell. Genetic material is enclosed in membrane in nuclear membrane in prokaryotic cell. Genetic material enclosed in a well defined nuclear membrane in case of eukaryotic cells. Genetic material consists of single circular or endless double helical molecule of DNA. In eukaryotic cells, there are many chromosomes formed of linear open ended double helical molecules of DNA. Now, in prokaryotic, DNA remains uncombined with protein. In eukaryotic, DNA remains combined with proteins. Next is fusion stain will not stain the chromosome, whereas in eukaryotes, fusion stain will stain the chromosome. Here, in prokaryote, nucleolus is absent, and in eukaryotes, nucleolus is present and it is rich in RNA. Centrioles are absent in prokaryotes, and centriole is present in animal cell but absent in plant cell. Cell division by binary fission, mitosis, and meiosis are absent in the case of prekaryotic cell, whereas in eukaryotic cells, division of somatic cells takes place through mitosis, but in reproductive cell, meiotic cell division take place. Now, student, the other is no spindle fiber formation during the cell division in case of prokaryotes, and in eukaryotes, spindle fibers form during cell division. The Golgi body is absent in prokaryotes, and Golgi body is present in case of eukaryote. Endoplasmic reticulum is also absent in prokaryote, and endoplasmic reticulum is present in eukaryotes. Ribosomes dispersed in cytoplasm in prokaryotic cells, and in eukaryotic ribosomes are present both in cytoplasm as well as on the surface of the endoplasmic reticulum. Next is, in prokaryotes plastids are absent, and in eukaryotes plastids are absent in animal cell except a few but present in the plant cell. Next, lysosomes are absent in prokaryotic cell, and in eukaryotic lysosomes are present in animal cell and the meristematic plant cells. Mitochondria are absent and the respiratory function is done by plasma membrane in prokaryotic cell. Whereas in eukaryotic cells, mitochondria are present and contain numerous enzymes, circular double helical DNA, and mitochondrial RNA. Next, in prokaryotes, the vacuoles are absent, whereas in eukaryotes, small vacuoles are present in animal cell and large vacuoles are present in the plant cell. Next, photosynthetic pigments are scattered in photosynthetic laminae in prokaryotes, whereas photosynthetic pigments are located in the plastid in the eukaryotic cell. This is the difference between the prokaryotes and eukaryotes.
Next, third question is, what would happen if the plasma membrane will rupture or it breaks down? So student, if plasma membrane breaks down, so there is a freely movement of substances. Okay, so the plasma membrane act as a barrier for the movement of the particles from into the cell and out of the cell. The movement of particles through the plasma membrane takes place by the processes of osmosis and the diffusion. If plasma membrane is not present, so there is a random movement of the particle and the substances dispersed all over the cell. Okay, so that's why the plasma membrane is necessary to maintain the equilibrium of the movement of the particles into the cell and out of the cell. The fourth question is, what would happen to the life of the cell if there is no Golgi apparatus? So students, the function of the Golgi apparatus is the synthesis of the substances and the packaging and dispatching of the substances. If there is no Golgi apparatus, so there will be no further process. Okay, so if there is no Golgi apparatus, various substances could not be in the position to be transformed in a proper use. Okay, if there is no Golgi apparatus, so the various substances are not in the proper position to be transformed in the further use. Okay, so there is a major role of Golgi apparatus. Clear? Next question, question number 5 is, which organelle of the cell is the powerhouse of the cell and why? Why it is the powerhouse of the cell? Students, as you know, the mitochondria is the powerhouse of the cell. Why it is called the powerhouse of the cell? Because it synthesizes the energy rich molecule, that is the ATP. It synthesizes the ATP, that is adenosine triphosphate. It is the energy rich molecule. It is the energy rich molecule. And it is synthesized by the mitochondria. That's why mitochondria is known as powerhouse of the cell. So the next question is, where do the lipids and protein constituting the cell membrane synthesized? So the question is asking that where is the synthesis of the protein and the lipids? Okay student, so their synthesis mostly occur in the endoplasmic reticulum. And you know, the endoplasmic reticulum are of two types, rough endoplasmic reticulum and the second one is the smooth endoplasmic reticulum. So the synthesis of protein takes place in the rough endoplasmic reticulum and the synthesis of lipids takes place in the smooth endoplasmic reticulum. So student, next question is, how does the amoeba engulf, student, next question is, how does an amoeba obtain their food? Amoeba obtain their food through the process of phagocytosis. Okay, so amoeba has an irregular shape It forms and temporary structures that are known as pseudopodia. Suppose this is an amoeba, okay, and this is an food particle. Clearly, so for taking this food particle, it forms the pseudopodia. These are the pseudopodia, the finger-like structure, and here is the food particle. So these structures are the pseudopodia. Clear? They are temporary structures and the food particles is here. After that, the food particles come inside the amoeba. Okay? The pseudopodia capture the food particle. So, it capture the food by forming the pseudopodia. Pseudo, the meaning of the pseudo means false. They are the false structure. After capturing the food particle, they disappear. Clear? You can see in this figure the process or the nutrition in the amoeba. Process of taking food in amoeba. This figure explains the process. Okay? So student, next question is, what is osmosis? We have already discussed it very clearly in the videos that the difference between the diffusion and the osmosis. Student, osmosis is the movement of the solvent from its higher concentration to its lower concentration through a special membrane. The membrane is present between the two solutions. Okay, so that membrane is the semi-permeable membrane or the selective membrane through which the particles moves from one place to another. Okay, so what is the osmosis? It is the transfer of solvent, the movement of solvent particles from its higher concentration to its lower concentration through a semi-permeable membrane. Clear?
Here you can define the osmosis in this way. Osmosis is the passage of water from a region of high water concentration to a region of low water concentration through a semi-permeable membrane. A membrane which permits the entry and exit of some material in the cell is called semi-permeable membrane. It also prevents the movement of some material. So this define the osmosis. Clear? Osmosis prevents the movement of some other particles. The selective particles are moved through the semi-permeable membrane. Here you can see with the help of this diagram also. In figure A, there is a concentrated sugar solution. The red color particles are of sugar solution, and next is the dilute solution. So here what will happen, the water from the part B starts moving into the sugar solution. Clear? So in the next figure you can clearly see the water molecules are present, are come in the section A. Clear? So there is an equilibrium maintained between both the chambers. Now, in this process, you can differentiate the osmosis and the diffusion. Here you can see clearly a semi-permeable membrane is present between the two solution. That is one the solvent and the other is solution. So this is a selective membrane that passes the molecules through it by their selection. Okay, the particles which require are only moved with the help of the semi-permeable membrane. Here you can say in the green particles are of solute. They are the sugar or salt solution. So there is a less amount of dilute. So the water molecules, the pure water starts moves towards the concentrated solution. So this is the osmosis. And in case of diffusion, the high concentration to the low concentration, the here the particles move randomly. There is no requirement of any kind of membrane. The particles moves randomly in the environment and they dispersed in their surroundings. Nutrition in amoeba. Amoeba eats microscopic plants and animals which float in water. The mode of nutrition is holozoic. In amoeba, nutrition carried out in 5 different steps just like the other animals. The first is ingestion, second digestion, third absorption, fourth one is assimilation, and the fifth one is egestion. So here the first step is ingestion. When a food particle comes near the amoeba, it ingests this food particle by using pseudopodia around it. Second is digestion. The food is digested in the food vacuole by digestive enzyme. Third is absorption. The digested food present in the food vacuole directly absorbed into the cytoplasm of amoeba by diffusion. Third is assimilation. Food absorbed by amoeba is used to obtain energy for maintaining life process. Last is egestion. When a sufficient amount of undigested food collects inside the amoeba, then the cell membrane suddenly rupture at any place and the undigested food is thrown out of the body of amoeba. This is the nutrition in amoeba. In amoeba, the digestion occurs within the cell. This type of digestion is called intracellular digestion.